Afternoon. I'm Kevin Brennan with the Notre Dame Alumni Association. Thanks for joining us once again for Behind the Headlines, the online learning series that allows you to engage with top Notre Dame experts on issues in the news. The latest outbreak of the Ebola virus has tragically claimed the lives of thousands in West Africa over the last several months, and the disease continues to spread at an alarming rate, raising worldwide concerns for the well-being of those affected in Africa and for hopes that we can develop a way to more effectively contain the spread of the Ebola virus. We're lucky to be joined today by one of the country's leading researchers on the Ebola virus. Professor Robert Stahelin is an associate professor of chemistry and biochemistry at the University of Notre Dame, as well as an associate professor at the Indiana University School of Medicine here in South Bend. And today he's joining us to share his expertise on the Ebola virus and give you a little insight into his cutting-edge research. Professor Stahelin, thanks so much for being here.
Thanks for the invitation.
For those of you watching at home, here's how today's program is going to work. Professor Stahelin is going to walk us through a short presentation on his research and the Ebola virus. And when he finishes his presentation, he'll be fielding your questions. You can submit your questions throughout today's program using the Q&A panel on the bottom right-hand portion of your screen, and we'll try to get to as many of the questions as we can as time allows. Professor Stahelin, with that, I'll hand it off to you.
All right, thank you very much again for the invitation. So I'd like to talk to you about the research we've been doing in my laboratory for the past 6 years or so on the Ebola virus. And I want to notify you with a disclaimer, we're not a high safety level lab. We're not working with live virus. Instead, we have models of the Ebola virus to understand how it replicates inside human cells. So on this slide, you see one of the early electron microscopy image of what the Ebola virus looks like. So you can see here it has an interesting shape. It's filamentous in nature, and it comes from the family of viruses called filoviruses. So really how I got interested in this virus was by reading the book The Hot Zone during my years in college on a fishing trip. I was just really intrigued by how this virus causes massive viral hemorrhagic fever, and of course, as some of you know from the current outbreak, has a high case fatality rate. So what my lab's been interested in seeing is how does this virus replicate in human cells? In order for any virus to sustain an infection, it has to enter the host cell, whether it's an infection in your pets or your own body. It then has to replicate. And during that replication cycle is really a novel time for which we can inhibit its replication and hopefully inhibit it with new therapeutics that may destroy the virus and hopefully then achieve a higher rate of cure. So what's known about the Ebola virus? From that slide I show you previously, we know, again, it's filamentous in shape, and it's very unique in the way it attacks the human body. So Ebola virus can enter most cells in the human body to essentially replicate or use those cells as a reservoir to make more virus. So the exact routes of transmission from animals to humans are somewhat well known and others not so much, depending on the literature you read. Our work is mostly focused on what's happening inside the cells of the human body. So early on in the infection, symptoms may appear anywhere from 2 to 21 days. So what's happening during this time? Well, what the virus is able to do, it actually can circumvent the immune system pretty well. It can infect cells in our bloodstream. It can then induce death to some of the helper cells in our immune system. So we have some cells called lymphocytes that never get infected with the virus, but instead the virus causes them to be turned off or essentially become inactive. This is a detriment because then what the virus does, it goes on to replicate in the lymphatic system, in the liver, the kidneys, and what you'll see on the microscopic level is you'll see leaky vasculature. So the capillaries of the body start to leak because the virus is infecting these cells. It's also affecting the way the blood coagulates. So eventually, of course, if not treated with supportive care, ways of replacing the electrolytes in the body or helping limit the way that the virus infects the immune system or the vasculature, unfortunately, we see a high rate of fatality, around 60% in the current outbreak going on in West Africa. So what's really interesting to me, because my background is not in virology, instead it's in chemistry and biochemistry, specifically looking at lipid membranes. So what are lipid membranes? Well, lipid membranes actually are necessary and fundamental to life. They're found in all cells in our body, and they really help give cells of our body stability and compartmentalize them. So shown here in this figure is just a cartoon of the influenza viruses. So many viruses in nature are actually lipid envelope. So they have a lipid bilayer that surrounds their genome. Okay, their genome is either made of RNA or DNA, and this encodes how they replicate, affect us in terms of our immunity, and so on and so forth. So we've been interested how this lipid envelope is derived in the virus. Where does it come from? And as it turns out, for these viruses that infect humans, the lipids actually come from our cells. Okay, so the current Ebola outbreak, the virus is infecting cells in the vasculature, for instance. It then replicates in those cells and actually hijacks or steals the lipids from the human cell that it infects. So we're trying to study how this happens and then use that as a therapeutic guideline to inhibit viral replication. So Ebola virus has not been studied in depth in terms of its replication cycle. Other viruses like HIV have. So one of the things my lab likes to do is to compare Ebola to other viruses in terms of how it hijacks the human cell. How does it utilize human proteins to essentially make a live and functional virus? So throughout the presentation, I'll compare a little bit of Ebola replication to that of HIV because it's more established, and there are therapeutics that can tackle and inhibit its replication cycle. So how do we study the Ebola virus in a regular laboratory, a laboratory that just has facilities to do tissue culture work and cell culture work of human cells, but not work with live virus? Well, the interesting thing about Ebola virus, it's filamentous, as I said earlier, and it's about 1,000 nanometers. This is 1 micron in length. What's really exciting about this virus in terms of how it replicates, its genome encodes only 7 proteins. Compare that to the human genome, which may have 25 to 30,000 different genes. It's quite remarkable that this simple genome is able to undergo a great replication cycle that increases the viral load in the body, and then, of course, eventually cause the patient to become very sick or unfortunately die. So of the 7 proteins of the virus, we've been studying what's called the matrix protein, and it has this acronym VP40 for viral protein 40, and 40 means 40 kilodaltons. This is the molecular weight of the size of that viral protein and how it got its first name more than a decade ago. What's exciting about VP40 in our eyes is this matrix protein lines the complete inside of the virus. So without VP40, the Ebola virus cannot replicate and form a stable viral particle. About 10 years ago, a number of researchers in the Ebola field demonstrated that just this one protein, VP40 alone, was sufficient to make what are called virus-like particles. You can see those on the bottom right of this screen. These virus-like particles are lipid envelope, and when first isolated, were almost indistinguishable from live Ebola virus. So this has served as a model for a number of different laboratories to study the budding and replication of the Ebola virus in human cells without the full intact genome that, of course, would be possibly lethal. So what I'm going to walk you through is how we think Ebola protein VP40 is regulating replication and forming the Ebola virus envelope. So once the virus is contracted, it may be through sharing of bodily fluids, the virus has to infect host cells to continue to replicate and make a person sick. So the virus does this through what's called attachment to a cell surface receptor. You can see that at the top of the screen here where it says attachment and endocytosis. Once the virus attaches, it then is taken up by human cells and it needs to fuse. So that lipid particle envelope of the virus has to fuse with human cell membranes inside the cell. Once this occurs, the viral genome is unleashed. So the virus then can propagate and create more and more of its genome, as well as more and more of the 7 proteins that are encoded in this RNA. Once these proteins are synthesized, they are trafficked to the plasma membrane. That's the inner leaflet here of the outer membrane of these cells. So down at the bottom left corner of this cell figure, you'll see a site that says assembly and budding. So what's happening here, this is the viral site of egress and budding, how the virus eventually forms new infectious particles. So this is the process we're studying. We want to understand how the virus forms new viral particles in our cells, which we hope to, of course, inhibit to dampen the infection. So to study VP40, we've done a couple of things, and we've been interested in figuring out how this protein functions in the human cell. HIV GAG is essentially the cousin of VP40. It's doing the same function in HIV infection, and from what's known in the literature on HIV, we've been able to start to understand a little bit on how Ebola VP40 is working. So HIV, shown at the top, has a site for which it interacts with the human cell membrane, the site of assembly and budding you saw in the last figure. This protein then can form these nice enriched ring structures, something called oligomerization. So these HIV gag molecules polymerize, and they form a shell that encodes the inner leaflet of this HIV virus. So Ebola is similar. Main difference is it's filamentous. So HIV is a spherical virus. Ebola, as you saw earlier, is filamentous in shape. And what VP40 does is it polymerizes underneath the plasma membrane of human cells, and it's specifically interacting with human cell lipids. And that's the question we're asking. How does it do this to achieve new viral particle replication? To first answer these questions, we took a look at what human cells look like in terms of what chemical lipids are there. So this is a cartoon of the cell, and you can see in this little box we've highlighted the plasma membrane where you have budding viral particles forming. What we know from about 3 decades of studies on human cell membranes is that there's different chemical components to membranes. And these chemical components are necessary for normal cell growth and division, but bacteria, parasites, viruses, what they end up doing during replication in human cells, they end up taking over these lipid pathways to achieve their own replication. So what we've been looking at is how the virus interacts specifically with these chemical components in our cells. Specifically, we've looked at some of the key regulators of Ebola virus regulation. So what we did first in our studies is we took the individual lipid chemical components of the cell plasma membrane and started to look at how they interacted with Ebola VP40 protein. So the main lipid I'm going to tell you about is shown on the top left. This is a lipid called phosphatidylserine. It's a fairly abundant lipid in all our cells. You see that about 20% of cell plasma membranes in human cells are actually made up of phosphatidylserine. So what we've been looking at is how this lipid interacts with the Ebola virus proteins. So the first way we did this is actually with a technique called surface plasmon resonance. This is a technique you can use to measure how strong two things interact from nature. So we've been looking at how the Ebola VP40 protein interacts with lipids from human cells. And this technique's nice because it gives us an idea of how strong the binding is. How tight does the viral protein bind to our human cell lipids? We then can compare it to our natural proteins in our body to see how VP40 may compete with human proteins for the same lipid pathway in order to achieve its replication. So the results for us were striking. What's shown down here on the bottom is just the chemical structure of this lipid, and what we think is happening is that VP40 can actually make chemical interactions through its sequence with the lipid head groups of this phosphatidylserine molecule. So what we saw, and these are called binding isotherms on the left and the right, we see that VP40 has what's called a very high affinity for this lipid that's found in our cell plasma membranes. It has nanomolar affinity. So very low concentrations of the Ebola VP40 protein are necessary to form new viral particles when it interacts with this lipid. So that data is shown here. We have a nanomolar affinity, and we compared that to some endogenous mammalian proteins shown as the bottom acronym in this figure. So endogenous human proteins that bind phosphatidylserine for our normal cell function have a similar affinity to Ebola VP40. So one of the things we think is happening here is essentially the Ebola proteins competing with human proteins for the lipid. That then means when the viral genome infects a cell and really upregulates the amount of that Ebola protein in the cell, it can outcompete natural human proteins for the lipid and then use that to form its viral lipid coat. So what's shown here is essentially how we study these things in live human cells. On the top left is electron microscopy picture from a publication that first identified that VP40 alone, so just one protein from the Ebola genome, can form these particles. So it's an electron micrograph of the plasma membrane of human cells, the outer membrane. And you can see these filamentous particles are forming and shooting off the membrane. So to study this in a time-dependent fashion, we wanted to ask the question, how long does it take for the Ebola virus to replicate and form new particles? What we did is we actually tagged our VP40 protein with different color markers. So there's a way to do this genomically. You can actually take DNA from a green fluorescent protein derived from jellyfish and put it in fusion with the VP40 protein from the virus. The nice thing then you get is this colorful protein that you can image with state-of-the-art microscopy. So we're actually able to see and observe the viral particles coming off the cell where Ebola is replicating and budding. So on the bottom left corner of this image, you can see the outer membrane ring is lit up by green. That's because the VP40 protein is found here after it infects the cell and then starts to form the virus-like particles. So we first compared VP40 to HIV gag. And the interesting results we saw early on is that they're very different in terms of how they interact with our human cells. VP40 is shown on the top, HIV on the bottom. And the green VP40, what we've done is we've merged it with different markers that can identify cellular lipid components. So VP40 and HIV are different. If you look on the right, VP40 has very little yellow color in this image. The yellow color is indicative of what's called co-localization, that the red signal and the green signal overlap. So the fact that they don't overlap demonstrates that VP40 isn't operating based upon how charged the membrane is. This is how HIV, shown on the bottom right, is highly yellow. That means it's operating on a highly charged interaction. The HIV gag is interacting through electrostatic charge with the poly-negative charged membrane. But Ebola VP40 is different than this. It's not interacting in this way. So then we went a little bit further in human cells to ask, how does this viral protein then sense lipids in the human cell? So on the top right, you'll see a merge that is yellow on the outer membrane. And this is a fusion image, really, of the green Ebola protein and a red endogenous protein that binds phosphorylserine. So indeed, VP40 does localize to where the phosphorylserine-rich regions of our human cells are. We also demonstrated that on the bottom right image. This uses an antibody. Antibodies are very effective because they can be generated to recognize just one epitope or one signal in a human cell. So we used a specific antibody for phosphorylserine, and on the bottom right we see these yellow patches wherever the green patches are for VP40. So what we've demonstrated here is that VP40 is using the phosphorylserine component of human cells to cause assembly and budding of new viral particles. We then went on chemically. We wanted to ask the question, can we use chemicals to inhibit replication and budding? So the first thing we did was more of a control and compared it to HIV. HIV can be controlled by neutralizing the charge of the membrane. So the plasma membrane where Ebola and HIV are binding is highly negatively charged. There's a lot of negative charge, and then the VP40 and HIV protein are positively charged. So you have what's called the charge-charge interaction that brings them together. Well, when you use this molecule called sphingosine, that actually neutralizes the charge of the membrane. So with VP40, we didn't see any effect. So charge neutralization is not an effective way at inhibiting its assembling and budding. For HIV, it is shown in the second image. So this really supported our PS story, where the PS is probably interacting with VP40, the phosphorylserine, and HIV is using more of that charge-charge interaction. So we then asked the question, can we inhibit the VP40 budding and formation of new viral particles by using endogenous proteins that bind phosphorylserine? So essentially, it's just an old competition assay, competing two things at different levels for the same binding partner. So shown here on the left is green VP40, and you can see compared to images I showed you earlier, there's very little ring structure around the outer membrane of these cells. Instead, when we overexpress or we upregulate how much endogenous PS protein is binding in those cells, it can compete with VP40. So we've been able to inhibit the way it interacts with the plasma membrane of human cells, and then inhibit the number of viral particles it can actually form. We also then asked the question, could we use antibodies to inhibit this process? Antibodies have been used now to treat a number of different diseases, often autoimmune diseases. A number of cancers can be treated with different types of antibodies. And now we're seeing more and more antibodies being engineered towards tackling viral infections. So what we've used here is an antibody that's been effective against HIV, and then that antibody towards phosphorylserine I showed you a few slides ago. So the HIV antibody wasn't very effective at inhibiting or altering any properties of the Ebola virus in terms of how it buds and replicates from human cells. So our control image is shown on the left, is labeled VP40. The center image, VP40 plus this antibody called a PIP2 antibody, is an anti-HIV antibody, and we didn't see quantitatively any effects from that antibody on replication. But we did by the phosphorylserine antibody. So when we actually use a phosphorylserine antibody, we were able to inhibit the virus from replicating and forming new virus particles. So this was very exciting to us, and we want to understand fully how this one protein from the virus controls its replication cycle. To further prove this out, we used a cell line that was deficient in the chemical component we think controls Ebola virus infection. So these cells are called PSA3 cells, and they're devoid of the lipid phosphorylserine in the cell plasma membrane. So on the left, what you see is that these cells do not support the normal ring structure of the green Ebola protein on the plasma membrane, nor do they support the viral replication. However, the neat thing about working with phosphorylserine is if you add it to the cells you're working with, they have enzymes on their outer membrane that will actually take the PS up and bring it into the cell. And when we add this to the cell, it actually reverses the Ebola virus budding. Under these conditions, we've restored that ring structure to the outer membrane in the center image, and we've restored the viral budding. This is also shown quantitatively on the lower left-hand bar graph. In our control cells labeled the CHOK1, we have budding from almost 100% of our cells. From our PS-deficient cell line, the replication goes down to about 30%. And then when we restore the chemical component of phosphorylserine, we've upregulated the budding and replication of the virus once again to about 80%. So we believe under these circumstances, we've been able to identify how the Ebola virus gets its lipid coat and hijack human cells to get this envelope enriched with phosphorylserine. So we're actually able to separate out these virus particles from the cell culture. You can purify them, and then you can study what's in the viral particles. So a couple individuals from my lab, Christina LeBlanc and Joanne Asquith, shown here, have worked out several protocols to separate the viral particles out from the human cells so we can characterize what chemical components are in them, in their lipid envelope, but also in their human protein contents. So viruses like Ebola and other viruses will actually steal some of the human proteins sometimes to also replicate to become fully infectious. And we wanted to ask the question how this happens. So what we also saw from these PS-deficient cell lines is we saw a great decrease in the number of viral particles that was formed. That's shown in this controlled bar graph. So in the CHOK1 cells, we have about 20 virus-like particles we see per cell that are formed. And then on the right, in the PS-deficient cell, we have less than 5 particles or so that are formed per cell. Okay, so this greatly shuts down the ability of the virus to replicate and form new virus particles. So what was really interesting to us is how does one viral protein form the viral coat? How could the virus encode just in its genome one protein to do this specific function? It must have an elegant means of interacting with the human cells. So we used what's called in vitro studies now to look at how VP40 is causing the plasma membrane of human cells to change its structure. So what's shown here in red are what are called giant unilamellar vesicles. These essentially are lipid membranes we make in the laboratory to recapitulate cells. So shown on the left is a control without the Ebola protein added. You can see it's red, it's spherical. We're imaging just one slice of a plane. Here we can actually image it up and down as well, but this is just one slice from the center of a spherical vesicle. So what happens when we add the VP40 protein to these membranes that recapitulate human cell membranes? Well, you can see in the second image from the left that VP40 forms these particles on the inside of the lamellar vesicle. So what we think is happening here is that this one protein binds to the outer membrane, it then deforms the membrane and forms these virus-like particles on the inside. So this is very much akin to what's happening on the plasma membrane of human cells when the Ebola virus is binding and pushing that membrane out to form the new filamentous particle. So we, of course, had to control this. We used PS binding proteins from humans and other animals. That's shown in the center figure, called LAC-C2, and that didn't affect the structure of the plasma membrane recapitulation at all, demonstrating the specificity for this viral protein in doing this to human cell membranes. We then went on to study and make sure that indeed this viral protein from Ebola was specific for this one chemical lipid entity from the human cells. So what's shown here is just a picture of different lipids we tested that are also found in the plasma membrane. And then on the right, we have a bar graph where we've quantified their contribution. So what we demonstrated all in all is that indeed phosphorylserine is the main component of the viral replication cycle that's needed to form a new Ebola particle. But this doesn't tell us really how chemically a protein is interacting with a human cell lipid. What we wanted to do a little bit further and better in terms of structural analysis is really visualize how does the Ebola protein specifically interact with a human cell lipid. This way we could then go back and design chemicals that could inhibit the Ebola replication better because we knew chemically how the two molecules interacted. So what we use to our advantage are a couple known proteins from humans that bind phosphorylserine. So fortunately, phosphorylserine has been what's called co-crystallized with these proteins. We have an image of how phosphorylserine binds, and we chemically know how proteins recognize the lipid. So we use these to compare phosphorylserine binding to Ebola to essentially understand out of the 20 amino acids in human proteins, which may be most important in Ebola VP40 for phosphorylserine recognition. So we did computational docking. We used a computer program to score and dock the places where the lipid most likely interacts with the Ebola protein. And what we identified on the right side, shown in green, is the lipid from human cells, phosphorylserine. Shown in the purple color are the residues that the computer demonstrated most likely interact chemically with the phosphorylserine. So these amino acids shown here are called lysine, threonine, and asparagine. And what we wanted to do is mutate them out. What if the virus no longer had these amino acids? Would that shut down this replication and inhibit its ability to bud from human cells? So shown here, indeed, these sites of interaction as predicted by the computer program seem to hold true in cells as well. So on the top left, you see the wild type VP40, again with this ring structure around the membrane. And then in the second one, you see one of those mutations. So we've taken out the lysine I showed you in the last figure. We've removed it. We've, put a different amino acid in. And we've been able then to show that no longer these ring structures form from the viral particle. Similarly, a second residue amino acid from that region also abrogated the ability of the viral protein to interact with human cell lipid phosphorylserine, as shown in the third image from the left. And then on the bottom, we were able to measure the amount of virus formed. So shown in the top series of bands is what are called an antibody for the virus-like particle. And what we show here on the right, you can see the lysine 127 and the threonine 129, as well as the asparagine 130, have knocked out the ability for the virus particles to form. So these residues in VP40 are essential to forming the replication cycle and interaction with human cell phosphorylserine. Last year, the new structure of viral protein 40 was published from Erica Ullman's Sapphire's lab at Scripps University. And her studies, were very interesting as they showed that this Ebola protein seems to form what's called a dimer. Two molecules of VP40 come together. So these two molecules are connected through what's called the N-terminal domain, shown in the blue color. It then has two free C-terminal domains, shown in the gold color. What their structural analysis also showed is that the protein eventually can what's called concatenate. It can form these long oligomeric structures that form a filament. So this is very exciting to us because we think that the lipid is what's doing the rearrangement. The interaction with phosphorylserine is essential for forming this long filament of the Ebola particle. So remember earlier I showed you a lipid envelope Ebola particle. Remember it was about a micron in length, and that VP40 protein was the most abundant protein in the virus particle, and it lined the complete inside of the virus. So this is how Dr. Saphire envisions that the virus forms. These VP40 proteins in the human cell eventually form a long filament that regulates the long budding filament that forms from infected human cells. Her lab then went on to show when you incubate VP40 with a lipid that mimics phosphatidylserine called dextran sulfate, it induces a great conformational change in the Ebola protein. So this backed up a lot of our work, demonstrating that phosphatidylserine was a key regulator of the assembly and formation of new viral particles. So we do something in our lab that physically and mathematically is very complex, but I think I can explain it in a very simple way. What we're able to do in the lab using state-of-the-art instrumentation here at Notre Dame and the IU School of Medicine South Bend is we're able to image single molecules that are moving in cells. So this goes back to that labeling of color that I showed you earlier, and we're using our green fluorescent protein tag to monitor how fast and dynamic the protein is moving throughout the cell. So we're actually able to count the number and brightness of the molecule. We can actually see the Ebola VP40 proteins interacting and see them forming these long filaments in real time in our cells. So this is exciting because we've been able to color code how the virus replicates and forms these long filaments that you saw in the structure a few slides ago. So this data has been published for a couple of years now, but what we've seen using what's called total internal reflection microscopy is that the Ebola VP40 protein assembles on the plasmid membrane as we expected in a phosphatidylserine-dependent manner. And what happens is you get formation of these oligomers very rapidly when it interacts with plasmid membrane phosphatidylserine. If we zoom in a little bit closer, we'll see in red on the right regions where VP40 has formed what's called the hexamer. This is where 6 molecules have come together to form a filament. And what we noticed early on is that these filaments were localized exclusively to the sites where the virus is forming. So our strategy now is to inhibit the replication of the virus by inhibiting its ability to form these filaments, or hexamers, 6mers, if you will, that form when it interacts with the phosphatidylserine. We've been able to do this through changing the viral structure. We can substitute the amino acids in the virus that form the hexameric region where it concatenates, and we can shut down the ability then to form new virus particles. So we can do it through mutation, and we're working on doing it through chemical therapeutics. So this led us to propose a model where the VP40 protein is in our cell, what's called the cytoplasm. This is the center of the cell, and there it's going to interact then with the plasma membrane phosphatidylserine shown on the left, and this interaction with the plasma membrane is what's responsible for inducing its filament formation into what eventually forms the virus particle. We went back then to our PS-deficient cell line to indeed show chemically that without this lipid in the cells, you cannot form the filament or the VP40 protein required for viral replication. So shown on the left panel is the cell line deficient in phosphatidylserine. Shown in the right panel is the cell line where we added phosphatidylserine back. So what happens on the left, we have very little hexamer formation as shown on the bottom. And then when we add PS back, look what happens to this plot of what's called frequency versus apparent brightness. We've greatly increased the number of VP40 filaments. And at the top of your screen on the right, you can see how you now have this ring structure highly localized to the sites of Ebola replication. But lipid binding often can be complex. It may not be as just simple as using one chemical entity in the membrane. So if you look across studies from the past couple decades, you realize that many human proteins, bacterial proteins, and viral proteins use more than one interaction to achieve their function. And this is something called coincidence detection, where it's detecting more than one component of the cellular membrane. So we wanted to make sure that indeed the PS binding doesn't induce binding to something else in the cells where the infection is occurring. So what we saw was interesting. It seems that there are complementary factors to the phosphatidylserine chemical in our cells. And these chemicals are called phosphonaphtalides. So what we demonstrated here using this assay, remember I call these giant unilamellar vesicles? And when we add the VP40 to the outside of the vesicle that recapitulates human cells, you get the budding into there of these viral particles. Well, when we added other chemical components that are found in small quantities in cell membranes, we found that a couple of these act as cofactors for viral replication. One of these is called PI45P2, the other is called PIP3. So we're interacting with probably phosphorylserine for VP40, which then causes it to interact with a second chemical entity. So we wanted to see if we could further inhibit the viral replication cycle by toying with the levels of these chemical entities in the cell. So we use a system created by an investigator at NIH named Thomas Bala, where essentially what you do is you use these components expressed in human cells to deplete the lipid chemicals PIP2 or PIP3. So we were able to do that by adding this rapamycin-inducible strategy system. And what this system does is it rapidly responds to the chemical agonist called rapamycin. It then causes a depletion specifically of the lipids I showed you on the last slide, the PIP2 or the PIP3. So we were able to use this system to demonstrate, do these other lipids act as cofactors for viral replication? And what we saw was very interesting. You have the chemical depletion system working on both the left and right image here. So on the left you see 0 minutes. That's before the chemical activation occurs. And then we have a change in response to 5 minutes of treatment. So after 5 minutes of treatment and depletion of these lipids from the membrane, the phosphorylserine, the PIP2, and the PIP3, we've caused even a more dramatic egress and replication of the Ebola virus than just phosphorylserine alone. So what we think is happening is that the phosphorylserine and PIP2 interaction, these phosphoinositides, are complementing each other in the plasma membrane to achieve the final step called viral scission. So for the viral particle to fully form, it eventually has to leave the human cell it infects. That requires a dramatic amount of energy for the membrane of our cell to actually break open slightly for a short period of time so the viral particle can leave and then the membrane reseal. So we think these cofactors of PIP2 and PIP3 are playing a role in the final step of viral replication where the virus actually leaves the human cell. So we can quantify this data as well, which is exciting for us because we see in the control image on the top, you can see that the gray and the purple bands overlap, and on the bottom image we see a great shift in the viral particle localization with respect to time. So when we deplete these chemical entities, we've greatly reduced the ability of the virus to form new particles. So the last thing I want to finish up with is talking about phosphorylserine as a cofactor for entry. If you remember early on, I showed you that phosphorylserine is localized on the inside of human cell membranes. But when cells become diseased or they die, they often cause the phosphorylserine to be exposed, and this tends to be a signal for their destruction. Last year, there was a very exciting paper published in the Ebola field, and these researchers demonstrated that phosphorylserine on the outside of the virus was responsible for viral entry, so essentially needed to infect more cells. And that, of course, was very interesting to lipid biologists because phosphorylserine is on the inside of the cell where VP40 is budding. How does it then get exposed in the live virus to help facilitate entry? So we wanted to ask the question, does this VP40 protein from the virus help control how the phosphorylserine is exposed and then used to enter cells? So we used something in the lab called flow cytometry, and this is able to count the number of cells that have a particular color. So what we have in green is a control of the green fluorescent protein. What we have in the left panel in gray is VP40 expressing cells. So what we saw using a label for phosphorylserine is that we get a great shift in the population of cells on the left from the green band to the gray band. And what this shift indicates is that phosphorylserine is indeed greatly exposed on cells expressing the Ebola virus VP40 protein. On the right is shown a control. So we use the human PS lipid binding proteins, express them in human cells. That LAC-C2 band is shown in purple and compared to the viral protein from Ebola VP40. And again, you see a great shift to the right comparing the purple to the gray. So that's striking that PS is exposed when VP40 alone is found in human cells. So it may control the way the PS is exposed in the virus that helps then facilitate later entry of the virus. We also became interested in studying this in other viruses because this same paper I alluded to in Ebola demonstrated that other viruses also use phosphorylserine as a mechanism of entry. So what we saw for HIV gag, it behaved a lot like Ebola VP40. Just one protein from HIV, shown in red, was enough to shift the curve and demonstrate that phosphorylserine is exposed in HIV gag-infected cells. And we have a control shown in blue demonstrating here no PS being exposed. Okay, but pictures are often better than these types of graphs, so we did imaging of single cells. So on the top, you have a picture of GFP VP40. You can see the green localization where the Ebola protein is, and then when we label PS on the outside, we see on the top right in the gray that indeed the phosphatidylserine is indeed exposed to the outside of the cell when the VP40 protein is present. So we've been able to quantify this, and we hope that in addition to inhibiting replication, that by targeting the phosphatidylserine exposed on the virus, we have another means of inhibiting its entry or infection in human cells. Okay, so in summary, what have I told you? Well, again, there's just 7 proteins in the Ebola genome, and one of these, VP40, which is the most abundant protein in the virus, is enough to make these virus-like particles. So what we've been able to show is that VP40 is a high-affinity PS binding protein. It has affinity for phosphatidylserine similar to that of our endogenous human proteins. So in human cells, it's competing with endogenous proteins for phosphatidylserine, which it then uses to regulate its plasma membrane-dependent assembly. The VP40 is able to do this by changing membrane structure. So it seems to be able to replicate by encoding in its own genome the ability, to change plasma membrane shape and to form these long filaments. Lastly, we see that phosphatidylserine is enriched in these particles, and then it's exposed only when VP40 is present. So VP40 causes PS to be localized in the virus particle, but also flipped to the outside and exposed as a cofactor for entry. Lastly, we think there's some complex chemical interactions happening, that the phosphatidylserine is inducing a secondary interaction with another chemical component called phosphoninositides, and we think this is what's controlling the final step of the virus leaving human cells to then go on and infect more cells to sustain the infection. So in terms of the acknowledgments, in this type of endeavor, you need a lot of good scientists, and that comes from undergraduates at Notre Dame, PhD students, postdocs at the IU School of Medicine. So my funding has come from the National Institute of Allergy and Infectious Disease. They've been very supportive of this work through our NIH R01 funding and supplemental funding, and then some of the other projects in my lab have been funded by the National Science Foundation, American Heart, and American Cancer Society. So I thank you guys for your time and look forward to questions.
Great. Thanks so much. For those of you watching live, you can submit your questions now. You can do so using the Q&A panel on the bottom right-hand portion of your screen there. To start off, we have a question I think that has been going around a lot in the news lately. There's been a lot of concern, and it's about the potential for the spread of the Ebola virus from West Africa to the United States. We've obviously had a few Americans come back and receive treatment here, but what do you think? Is that a serious risk? Is that something Americans should be concerned about?
I don't think it's something that someone should be majorly concerned about. It certainly can happen with the current state of travel. Travel pattern predictions would indicate that certainly the virus could come to the United States through airplane or other means. But containment facilities, recognition of disease, the way we respond to disease is much stronger than other countries. So in terms of being a severe risk at this time, again, that's very low.
And how exactly does the disease spread from person to person?
Great question. So this is going to require direct contact with bodily fluids, vomiting, diarrhea, sweat, saliva. These types of fluids, of course, can spread the disease. It's not an airborne virus. Obviously, hygiene is very important, so one has to be very mindful of that. But essentially, you're going to need direct contact with bodily fluids to actually acquire the viral load to infect your body.
The experimental drugs which were used to treat patients, infected patients at Emory Hospital in Atlanta, has gotten a lot of attention in the news. Is there any overlap between that and your research? Does it draw on any similar theories? Are they related in any way?
So the ZMapp is a cocktail of antibodies. So they're really related in the sense that we've been using antibodies to inhibit the virus, but what the antibodies do in recognizing the virus are much different. So the type of chemical entity is similar, but the way it's inhibiting the virus is different from what we've been studying.
And the research that you were bringing to us today involving replication that you're studying, is that something that how can that go from research to maybe some sort of drug therapy which can help treat the Ebola virus.
That's right. So a number of antiviral drugs on the market these days, HIV inhibitors, herpes simplex virus inhibitors, cytomegalovirus inhibitors. A lot of these drug therapies came from this type of basic science approach, and that is indeed studying how the virus replicates in human cells. So some of the classical drugs to inhibit virus have been what are called DNA polymerase inhibitors. They inhibit the ability for the virus to replicate its genome. So what we're doing here is we've been screening for chemical molecules. What I didn't show you is been working with a researcher from Europe and a researcher from Canada in the lab where we actually have chemicals where they've synthesized to inhibit the VP40 polymerization. So we hope that these molecules, they work in cell culture for inhibiting the virus. The biggest problem with drug development often is toxicity. How toxic going to be to the host cell? So we hope in the next 5 to 8 years to have some molecules that would be useful for inhibiting virus, at least in animal studies.
And we have another question about kind of the treatment. It seems that obviously you read all the news stories about how deadly and how quickly it can be deadly in Africa, but the folks who've come back to the United States, so far this experimental treatment seems to have succeeded somewhat, or at least, temporarily succeeded. What are the major differences? Is it just this experimental drug treatment? Could that be brought over to Africa and achieve similar results, or are there larger problems that would make it more difficult?
Yeah, I mean, logistics is important. I'm not a physician, I'm a PhD, but I mean, my concern with drug efficacy in third world countries is what type of supportive care can the patients get? So we know, for example, you alluded to Dr. Kent Brantley, who was IU School of Medicine graduate. He was on a mission trip serving these patients with Ebola, and they brought him back to Emory University for care. So in that type of environment, the care he got was very, very, very important. Electrolyte replacement therapy had to be very aggressive to essentially keep up his normal bodily function, the liver function, the kidney function, what's his blood pressure. This is going to be important factors for complementing the therapeutic. So therapeutics may be able to inhibit the virus, but you still have to make sure that the patient hasn't succumb so much to the disease internally where their organs are starting to fail, because that eventually happens in the Ebola infection.
Reminder to those of you joining us live, you can submit your questions now using the Q&A panel on the bottom right-hand part of your screen. We have one question about your background. How did you first become interested in studying Ebola, and kind of how did you come to this research?
Yeah, so the first thing, what I tell undergrads and grad students is that get a good foundation in education so you can eventually branch out and do really whatever you want. So my PhD is in chemistry, my undergrad's in biochemistry, and before coming and becoming an independent faculty member, I hadn't touched anything in virology. I never worked with a virus at all. This book that I read, and I think it was around 96 or 97, I read it during a fishing trip in college called The Hot Zone by Richard Preston. It talked about the Ebola Reston outbreak in Reston, Virginia, essentially where rhesus macaque monkeys were brought in from the Philippines, and they had a strain, or a species, excuse me, of Ebola called Ebola Reston, which caused the monkeys to die, but the humans luckily never got sick. So that was very intriguing to me. How did this virus replicate? How did it essentially shut down, at least in that case, the primate immune system so much that they died? So when I started my own lab, I started looking for really diseases that didn't have much basic science done on them, and Ebola was one of those. So because its genome was simple and we could study just a couple proteins outside the live virus in a BSL-2 lab, a biosafety level 2 lab, I applied my background in chemistry and biochemistry and then these lipid membranes I talked about to really get a foothold on how this virus we think is forming these particles.
We have a question here from a viewer who's a clinician, and she says that she's curious how long some of these questions will take to answer. How hard is it to color all these particles and visual, and how many trial before you can have success with these new studies?
Yeah, success towards human therapeutics is usually years. Success towards getting publication and validation, where this becomes kind of a textbook answer to how it replicates, is a little bit shorter. So I know working with some other people from other universities who've been studying VP40, they at least see similar types of data from what we've seen. So we're pretty confident the replication cycle in terms of this lipid interaction is controlling the regulation. But I mean, we're at least 5 to 7 years away from therapeutics stemming from my laboratory being useful in humans. Again, they've worked in cell culture, so we see little toxicity in human cell lines for inhibiting the virus, but we don't know how they're going to interact when the body metabolizes them or how they interact with different organ systems.
And we have another question about the situation, the current outbreak in Africa, which is is there a solution kind of on the horizon to stop the spread and work towards containment
Yes.
in West Africa?
Yes, I know at least the World Health Organization is estimating obviously a large number of cases and predicting somewhere around a 6 to 9 month type of time frame for full containment. I've never worked myself in this type of logistical fashion in terms of trying to contain something of this nature. So I know that the urban environment being, and also this virus being found in different countries, has really made it difficult to fully contain it very carefully. But it seems that they eventually may work towards that on a fairly reasonable time frame, I would say, for the amount of cases that has been seen. But I can't speak myself to what is needed. I know Doctors Without Borders and a couple other organizations are calling for, obviously, volunteers, fundraising efforts, as a lot of outbreaks or hurricane disasters, things like this we've seen in the past. The more people that can get involved, either financially or helping, that's very important. Obviously, here we have a different situation with this very infectious agent. But I think sometimes more than just people on the ground can be helpful in getting support and containment.
Great. Reminder, we have a few more minutes for questions, so if you have some questions, please submit them now using the Q&A panel. Excuse me. We have a question here about the future of your research. Where do you see the research going over the next 5 to 10 years?
Yeah, so we become very excited about how this lipid is exposed, this phosphatidylserine is exposed on the VP40 expressing cells. And not just maybe for Ebola, but for other viruses as well. Because we saw this with HIV, and we're working with a couple other viruses now that likely somehow cause the phosphatidylserine to be exposed on these cells that eventually become part of the viral particle and help facilitate entry. So a lot of our work is going, we're still studying Ebola. We've actually moved over the last 3 or 4 months. A new graduate student in the lab has been studying what's called Marburg virus. Marburg actually is a filovirus family member that was discovered before Ebola. It was first discovered in 1967, and it has a high lethality rate as well. In fact, some of its outbreaks have been more lethal than that of Ebola. So we're excited at comparing Marburg and Ebola because they seem to be different. That VP40 protein from Marburg is shorter, it's not as long in its amino acid sequence, and it doesn't have as much homology. So we want to see how these two filamentous viruses may differ in forming new viral particles.
There's been obviously a ton of media coverage about the Ebola virus. What have you seen in kind of the coverage in the discussion about the virus and the threat that it poses? What are the biggest misconceptions that are out there currently about the Ebola virus?
Yeah, I think some of the misconceptions are essentially the rate of spread and the type of spreading. And obviously, again, through the direct contact with bodily fluids, that obviously doesn't mean direct contact with someone. If I obviously spit or something on the ground and someone touched it, this still could be virus that you can get it from. So I think sometimes a lot of people believe that it's transmitted much more easily than it thinks. It's not a very stable virus. So that's probably the most common misconception I've seen that I can think of.
Okay. And we have a question here about the role that you mentioned some Notre Dame students were involved in the research, and one of our questioners was curious to learn more about what role that they've played, the students have played.
Sure. So a number of undergrads have worked on this project. Let's see on the pictures up there. I don't have all the updated pictures of all the undergrads. A couple undergrads have published papers on how this VP40 protein from the Ebola virus is working. So they're usually paired with a graduate student or a postdoc early on in the lab, and then we really work to have them become somewhat independent. Of course, their time is limited compared to a grad student. They may work in the lab from 10, 12 to 25 hours a week, usually over 2 or 2.5 years. Try to get them in their sophomore year or right before their junior year. And then sometimes they work with funding over the summer, which is nice. But they eventually become almost like a functioning graduate student. We have them do some of the live cell imaging you've seen, some of these measurements of how the protein's interacting with the chemicals from the lipid membrane. So they play really an important role of progressing the research. And then it's important for education and outreach as well, because if we can get more people studying this type of basic science and get them going to grad school and med school, we'll have a better force to study these types of things in the future.
Great. Well, Professor Stahelin, thank you so much for joining us today, and thanks for all the work you're doing, the research you're doing to help fight this deadly disease. We really appreciate your time.
Thanks for your time, and thanks for listening.
And thanks to all those of you who watch live at home, and to those of you watching on our YouTube page later. If you'd like to keep up to date on our upcoming online learning events, please go to my.nd.edu/online learning. You can also watch all of our previous online learning events on our YouTube page. Just search for the Notre Dame Alumni Association on YouTube. I'm Kevin Brennan with the Notre Dame Alumni